The central dogma of molecular biology encompasses DNA, RNA, and protein synthesis. It states that DNA makes RNA, which makes protein. So ultimately, DNA is processed into RNA through the process of transcription, and RNA is processed into protein through the process of translation. But for this video, we'll focus in on the first step, called transcription. So transcription is the process by which DNA is copied, or another word is transcribed, into mRNA, also known as messenger RNA. And mRNA carries the information needed for protein synthesis. But first, pre-messenger RNA is formed, with the involvement of RNA polymerase enzymes. The RNA polymerase enzymes uses a single-stranded DNA template to synthesize a complementary strand of RNA. It builds an RNA strand in the 5' to 3' direction, adding each new nucleotide to the 3' end of the strand. And there are 3 stages of transcription: initiation, elongation, and termination. In the initiation stage, RNA polymerase binds to the sequence of DNA called a promoter that's found at the beginning of a gene. So each gene, or often a cluster of genes, have their own promoter. But bacteria and eukaryotes have different types of promoters. So a typical bacterial promoter has 2 important DNA sequences called the -10 and -35 elements. RNA polymerase recognizes and binds directly to these sequences. The sequences position the polymerase in the right spot to start transcribing a target gene, and they also make sure that it's pointing in the right direction. Once the RNA polymerase has bound, it can open up the DNA and get to work. So DNA opening occurs at the -10 element, where the strands are easy to separate due to the many A's and T's, which bind to each other using only 2 hydrogen bonds rather than the 3 hydrogen bonds of G's and C's. But this process is a little bit different for eukaryotic cells, like human cells, for example. In eukaryotes like humans, the main RNA polymerase in your cells don't attach directly to the promoters like bacterial RNA polymerase. Instead, these different helper proteins called general transcription factors bind to the promoter first, and this helps the RNA polymerase in your cells get a hold on the DNA. Many eukaryotic promoters have a sequence called a Tata box. The Tata box plays a role much like that of the -10 element in the bacteria. It's recognized by one of the general transcription factors, allowing other transcription factors and eventually RNA polymerase to bind. So in the next step, we have elongation. This is where one strand of DNA, called the template strand, acts as a template for RNA polymerase. So as RNA polymerase reads this template, one base at a time, the polymerase builds an RNA molecule out of the complementary nucleotides, making a chain that goes from 5' to 3'. The RNA transcript carries the same information as the non-template or coding strand of the DNA, but instead of thymine, there's the base uracil. Finally, there's termination. In this step, sequences that are called terminators signal the RNA transcript that it's complete. Once they are transcribed, they cause the transcript to be released from the RNA polymerase. So in the example shown in this video, a type of termination mechanism involves the formation of a hairpin loop in the RNA that's shown here. After termination, the RNA transcripts are immediately processed and ready to be used as messenger RNA in bacteria. For eukaryotes, on the other hand, there's more post-transcriptional processing involved. So right now, it's only going to be called pre-mRNA. And to be fully ready for translation, we need a few more steps. First, the pre-mRNA needs to have their ends modified by adding a 5' cap at the beginning and a 3' poly A tail at the end. Eukaryotic pre-mRNAs also undergo splicing. So this is where parts of the mRNA, which are called introns, are chopped out, and the remaining parts, called exons, are stuck back together. Genes aren't transcribed all the time because transcription is a highly regulated process for each and every gene. Cells carefully control the gene to be transcribed depending on which products are needed at a particular time. So for example, we can see here that gene 1 and gene 2 are both transcribed, but gene 1 undergoes transcription more due to the increased amounts of mRNA products present. So I hope that gives you a good overview and summary of the process of transcription. And now we move on to the next step, known as translation, where the mRNA sequence will then be used to synthesize polypeptide chains that can then be used to make protein.